This is Nervous System Part 2, How a Neuron Works. You watched the overview of the nervous system prior to this. This will probably be divided into two videos, but let's get started. And I apologize for the sound of my voice. I've been really sick, so if it starts to get raspy by the end of this video, that is why. Again, this is our overarching theme if you want to read this again. And we've seen this image before. This is the pathway that we will always see in the nervous system. Also, you should know the parts of the neuron at this point. This is a really cool video to look at the conduction of an electrical impulse if you want to. It's on YouTube. So when we're talking about cells, what we need to realize is that there's all sorts of ions inside and outside of a cell. Ions like sodium, right, Na+, potassium, which is K+, chlorine, which is Cl-, and it's just salts, right? It's like electrolytes that we consume in a Gatorade. There's naturally salt in our body as well, and those create charges. Depending on how much is inside the cell or outside of the cell, I'm going to see what we call a concentration gradient or an electrical gradient. Basically, more positives on one side and more negatives on another side. When a nervous cell is at rest, so it's not sending any message, it is more positive outside of the cell and negative inside of the cell. It's actually about -70, -75 inside of the cell. That means that all of my sodium, all of my potassium, a lot of that is outside of the cell, and I actually have a lot of chlorine, which is negative, inside of the cell when my nervous system is at rest. We can actually see that, excuse me. What we see is that a lot of my potassium is outside of the cell, which makes it very positive. I do have a lot of sodium inside of the cell, but what negates that is the chlorine that's present as well, which ends up making it a little bit negative. When we start talking about how a message moves down a neuron, we want to talk about the idea of an action potential, which is basically an electrical impulse that carries the message along the axon of the neuron. What happens is that the membrane of the neuron polarizes by changing that electrical potential or the positive-negative balance from the inside and outside of the cell. In response to a stimulus, so it's always going to be a chemical stimulus like a neurotransmitter, that's going to hit the dendrite and go to the cell body. What ends up happening is sodium, potassium, these channels open, and it causes the membrane to become depolarized, or it starts to make the inside of the cell become very positive. We know it is very negative when it's at rest. This is powered by ATP, and so we also need energy to be able to conduct these electrical impulses. This is basically how an electrical impulse proceeds. The axis on the left is actually going to be your charge. Down here in the red, it's about -70, and it's going to go up to 0, and then all the way up to the white line, which is very positive. And we'll talk about what each part of these colors mean. What's happening inside of the cell at each of the phases of this electrical impulse. Basically, we're looking at how a message is sent. The first part, which is going to be that red baseline, this is basically resting potential. What's happening here is most voltage-gated sodium channels are closed, so sodium is not moving back and forth. Most of the potassium channels are also closed. And so we're kind of keeping a very normal balance, negative inside, positive outside. Part 2, which is going to be my little blue bump right here, means that the voltage-gated sodium channels open first, and sodium flows into the cell. And we see that my cell starts to get more positive. Still negative, but we're adding positives to it, so it's approaching 0. This is called the rising phase, and then once the threshold is crossed, it's going to increase and go past 0, but it has to get through this rising phase. And again, we see that we started out at about -70, -75, and during the rising phase, I have sodium coming in, and I start to get less negative. During part 3, we see that like rising phase. So part 2 is going to be the blue, and then 3, the dark blue, is actually our rising phase. Rising phase, still tons of sodium being dumped into the cell, making it less and less negative, and eventually once it crosses 0, it starts becoming very positive. We get our green line, which is the falling phase. During the falling phase, the voltage-gated sodium channels close again, and the voltage-gated potassium channels open, and potassium starts getting dumped out of the cell. And so as I'm losing positive charge inside the cell, what do we see? It starts dropping in charge again, and the inside of the cell starts getting negative again. We also see something called an undershoot, which is going to be this little purple dip. During the undershoot, what ends up happening is more potassium than is necessary is pushed out of the cell. So it actually gets more negative than it needs to. A little bit of potassium flows back in, and it regains its original negative balance, but it kind of like bottoms out and then goes back to normal. Again, at 1, I'm at resting potential again. We also see that we have a refractory period, which is basically when a second action potential can't be initiated. The refractory period is a result of a temporary inactivation of the sodium channels. So basically that rising phase cannot happen again for a certain amount of time. It prevents the cells from becoming exhausted. How does this action potential get conducted? Essentially what happens is at the site of the axon hillock, which is the connection of the cell body to the beginning of an axon, an electrical current depolarizes one area, and then it starts moving along the axon, and it only moves in one direction. It moves from the cell body towards the synaptic terminals. The reason that this happens is because we have this refractory period where the inactivated sodium channels can't open for a certain amount of time, and so the cell remains depolarized in that area, making the impulse only move in one direction, which is actually a really important thing. This is what it kind of looks like, right? I see that the blue is the depolarization, and the action potential, that is the signal moving. It's then moved one spot to the right, and what is green in part 2 actually cannot be depolarized again because there's too much potassium outside of the cell, and then we see it move along like this. Try to sequence these, see where you're at. This is them sequenced, so I would pause if you want to check your work. Also, make sure you are able to label each of these parts and explain what is happening with the ions at each of these parts. Check your knowledge here. Check your knowledge here too. We can go over these answers if you'd like to. And check your knowledge here. There's a little bit of evolution review. This kind of answers evolutionary adaptations of the axon structure. As my neurons get bigger, the speed of my message gets faster, so that means that larger organisms potentially have faster impulses. In vertebrates, we have a myelin sheath, which increases the speed of the action potential as well. Myelin sheaths are made by glia in the central nervous system and by Schwann cells as well. We've talked about these a little bit, right? These is insulating cells that propel the message forward faster. What actually happens is that the message does not get transmitted through the Schwann cell. It only occurs at the nodes of Ranvier. And we will talk a little bit about this in the next set of slides.